use some of those chemicals for energy sources, or they use light for energy. They transform some of those organic chemicals, for example, into different building blocks that now are used to make the macromolecules, and the macromolecules comprise the cell. So all those chemical reactions occurring, somewhere around 3,000 chemical reactions predicted from just a little Escherichia coli, okay, to have it grow and reproduce. Microbes are able to grow and reproduce, so all of these cells are able to grow and reproduce. They take chemicals from the environment and they make new chemicals, new macromolecules. In a bacterium, you have one cell, it's carrying out metabolism, increases, almost doubles in size, and then it divides and gives you two cells. That's something we're going to learn. It's called binary fission. Once again, this isn't on the PowerPoint, so you know I'm not going to ask you binary fission on your first test, because we will not have gone to growth and reproduction. We're just trying to give you a peek of what's going on in the future as we get into the other concepts in the course. It just shows you some ancestral cell and what time it has changed, and as more and more changes occur, they can become distinct species. So metabolism, growth, evolution, all properties of all living cells, at least the potential for that. And there are some properties that are not found in all cells, but they're characteristic of some cells. Things like motility. You see a little bacterium here with this little long flagellum here. That happens to be a little polar flagellum. It's at one end. And so it's going to rotate. In fact, it's not wave-like motion like eukaryote. It rotates like a propeller and moves that microbe through its environment. Some bacteria and some cells have the ability to differentiate and make a new cell structure. This is one time I don't agree with the book fully. I don't like the term differentiation to describe the formation of an endospore in a bacterium because that's just making a special structure. What I usually like to reserve the term differentiation for is when you end up having, for example, an egg be fertilized by the sperm. And now you go through all those mitotic, the first few divisions, those cells are almost alike, almost at base. It's what happens as you get multiple mitotic divisions and the slight changes in the environment and the turning on and off of specific genes that eventually, as you get further mitotic divisions, those cells specialize. That's why you and I have intestinal mucosal cells that help transport nutrients across our intestine. That's why we have skin cells for rigidity to prevent microbes from penetrating. That's why we have kidney cells that are used in detoxification. All those cells have changed. Certain genes have been turned on and certain ones have been turned off, so that each cell has its own unique complement. Those cells have what we said differentiated into cells that have specialized function. So I like to use differentiation for that process whereby those cells become specialized. Okay? As far as this, I'm just saying some cells have the ability to form an endospore, not many, but I wouldn't want to call it differentiation. Yeah, it changes, okay, but differentiation I think is more appropriate for specialization. Okay, cells can communicate typically by chemicals, and we may talk about chemotaxis and movement of cells toward chemical gradients a little later on in the course. Okay, we've covered that, make sure all your notes. Okay. There's another thing, a way we can look at cells. We can look at cells in two different ways. As machines, we're going to look at catalytic functions, and we can look at them as coding units, genetic functions. So there's a little diagram. Let's go back 10 minutes here. So we can look at cells as chemical machines. What they're going to do is they're going to take an energy source in, they may oxidize it, they'll carry out all kinds of metabolic transformations. They'll generate energy from that energy source. So basically, we can talk about them being chemical machines, and there we're really talking about the catalytic functions, because those reactions would not occur in living cells if it weren't for the presence of enzymes. Living cells typically live at temperatures at which chemical reactions would not occur at significant rates. They would occur if we waited long enough, okay? But we'd be waiting centuries almost for to take in glucose and completely oxidize it, unless there was something to provide the energy of activation. So that's why we have to have enzymes. Enzymes catalyze the reaction. So when we look at the chemical machine function, that's dependent upon the presence of biological catalysts or enzymes. Also, we can look at cells as coding devices because all the properties of the cell are encoded in the double-stranded DNA molecule. And that code is transcribed into a messenger RNA, which then translated into polypeptides. So we can say that they're coding devices. This basically is the genetic function, analogous to computers, which store and process genetic information. That genetic information is in DNA, and eventually it's passed on during replication. So DNA serves as a template for its own replication. DNA also stores the genetic information in one of its strands for the synthesis of messenger RNA. And not only that, it also codes for ribosomal RNA sequences and transfer RNA sequences. As well, the messenger RNA can be translated into specific polypeptides. Okay, so here's a little diagram. Here's the genetic or coding function. DNA, double-stranded DNA, serves as a template for its own replication. Okay? And DNA, and actually it's one strand of DNA, is transcribed into messenger RNA. You can also transcribe it into ribosomal and transfer RNA, which are needed for the messenger RNA to become translated into the polypeptides, which comprise the proteins of the cell. So all of this is part of the coding function, okay? Or what we call the genetic function. And once translated, then these proteins, some of them serve as the enzymes that are needed for the catalytic function. So these two are completely separate. There's a link. The enzymes link the two together. The product related to the coding function, the enzymes or the polypeptides that serve as the biological catalyst to carry out those metabolic transformations. So these catalytic functions generate energy in the form of ATP. In metabolism, we carry out a series of reactions that biosynthesize all the building blocks needed to make the macromolecules, because the macromolecules are basically the major constituents of the living cell. Our proteins, our nucleic acids, our polysaccharides, and our lipids. 96% of the weight of a cell are those macromolecules. So the catalytic function then is to use the energy that is as a result of metabolism, as a result of the catalytic function, and continue that catalytic function to make all the precursors of the major macromolecules. So these two are linked here. There's the product of the central dogma of molecular biology. The central dogma of molecular biology, we'll see this again. DNA serves as a template for its own replication. DNA serves as a template for RNA, and RNA is with transcription, and RNA is translated into polypeptide chains as part of the translation process. Okay, this is just a little diagram. I'm not so interested in knowing all the details of the years, but basically, if you look at the origin of the Earth, somewhere about 4.6 billion years ago, they estimate, by all the best evidence they have, about 3.8 billion years ago, the origin of cellular life. That origin of cellular life was microbes. Okay? And the environment there was such there was no oxygen. Okay? So these had to be what we call anaerobic, or if you want, anoxygenic. So and because the sun was there, most of these were phototrophic. They were anoxic phototrophic bacteria. So the Earth was very limited. They had no oxygen there. Eventually, the cyanobacteria evolved pretty close to about 3 billion years ago, and the cyanobacteria could make the oxygen, so the Earth was slowly oxygenated. And if you look, eukaryotes first appeared about 2 billion years ago, right in here. Those some of the very early ones, the nucleated cells. And it included both microbial and multicellular right in here. Okay, and then, eventually, as that oxygen level gets high enough, then, the algae brought the oxygen level with algae even higher. So it wasn't until really late that we ended up getting the oxygen levels. So around 500 to 800 million years ago, the current oxygen levels were achieved, and then we could start having organisms that required oxygen. And you see, basically, if we look at our history, we're right here. And in fact, if we look at the history of the Earth, it basically 80% of the Earth's history was exclusively microbial before anything bigger than those microbes ever appeared. So if we look at your book called Luca, The Last Universal Common Ancestor, okay, that's where bacteria first split from eukaryote, okay? And then it was later that the archaea split off from eukaryote. So these are the 3 domains of life that we'll talk about later on, and we'll talk about Wose's work in chapter 2. So just give me a little hint of it there. Okay. Distribution of microbes on the Earth. Well, the great majority of microbes are in the marine sediment. The marine sediments cover more than 2/3 of the Earth's surface, okay? And they've been estimated to contain as much as 1/3 of the Earth's prokaryotic biomass. So we have the marine subsurface, okay? But if we actually look at the sediments, about 1/3 of the bacteria there. If we look at the full marine subsurface, about 66% of bacteria there. If we look at the soil, if we look below the surface of the soil, the subsurface, 26% are there. We look at the very surface of the soil, where there's a little bit of oxygen, about 4.8%. Oceans, 2.2%. See, I would have thought the oceans had more, but it's the sediments in the oceans that have most of it. And then there are other habitats they list here, for example, freshwater salt lakes, domesticated animals, ice, termites, humans, and domestic. Even though we have 10 to the 14th on our body, we're still tiny. It's a tiny number compared to all the microbes in the world. So microbes do play tremendous numbers on this earth, and most of them are in the marine sediments. We can talk a little bit about history now, and what I'm going to talk about history from the perspective of how did microbiology develop as a scientist? How do we learn about microorganisms? How can we study microorganisms? Because before 1850, very little was known about microorganisms. Bacon in the 13th century, he was more of a philosopher than a scientist, talked about these little invisible animalcules responsible for infectious or contagious diseases that spread through populations. But you know, he was just postulating something I don't know about. We'll call them invisible animalcules, okay? Because they thought they had some kind of special way of causing disease. And he was right. There were invisible microbes that, at least not visible to the naked eye, that were responsible for contagious diseases. But that was all speculation. We did not see microorganisms. In fact, if we look at the early history, 1684, Anton von Leeuwenhoek was the first observation of bacteria. Okay? Now, what he looked at was less than you see in the laboratory. And in fact, when you guys go to the laboratory, what have you done this week? Just looked at negative, did you do a negative stain? Yes. Yeah, you see a round or a rod or whatever, that's it. When you do a gram stain, you'll see a pink circle or a blue circle or a blue rod. Do you know that probably after next week, you probably won't use the microscope except again when you do unknowns? We're going to do very little with microscopy. How many of you, like me, when I first took the course in microbiology, expected to go into a microbiology lab and look into the microscope the whole time? Raise your hand. I was the same way. Okay. Disappointment is going to occur if you really want to do that, because in that bright field microscope, we're going to talk today about how limited we are with it. We really can't see much. We can do a gram stain, divide all bacteria into gram positive that stain blue, retain the crystal violet iodine complex, and those that are gram negative that lose that dye and get stained by a counter stain. We might be able to see an endospore. We can't see the flagellum or the bright field, but we can precipitate dyes on it, maybe. We're not going to do this in micro because it's hard to get it to work. You get one student out of 48 will get it to work. Precipitate the dye so that it's thick enough that you can see the precipitated dye. You don't really see the flagellum. Actually, to see anything internal to the cell, pretty much, with the exception of the big endospore, you've got to go to electron microscopy. And we don't have an electron microscope in the general microbiology lab for you to look at. But there you'll see ribosomes. If it's a transmission, you might be able to see the ribosomes clearly and so on. We'll show you some pictures of electron microscope. Well, Leeuwenhoek's microscope was a single lens system, not anywhere near good as the microscope you had, so microbiology did not develop in 1684, even though a lot of people want to call him the father of microbiology. 1798, Jenner, gosh, we get vaccination. He developed the smallpox vaccination. You think, well, wow, he knew about microbes. He didn't know what caused smallpox. All he knew was that milkmaids that would milk the cow, if they got this disease called cowpox, they had these little cowpox pustules on it that they got from whatever that invisible thing was that was causing cowpox. He noticed that those milkmaids that had the cowpox pustules, when there was a smallpox epidemic, they never came down with smallpox. So he theorized there's something in the cowpox pustules that make them immune to smallpox. And so what he did is he deliberately exposed the little boy at a time when there was a massive outbreak of smallpox. This young child was going to be exposed anyway. He deliberately exposed him after he inoculated him with cowpox pustule material from the milkmaid. And the young child did not develop smallpox. So that was how the first one, but he didn't know what caused smallpox. Okay? So we still didn't develop microbiology, even though we had vaccination. Here's when we start getting at the tools we need to study microbiology. Pasteur was involved in the controversy about living organisms arising from non-living matter. Spontaneous generation was a theory that you could take non-living matter and you could have microbes spontaneously arise from non-living matter. And where they got that idea is if you had any kind of nutrient material, even meat out, if you leave it long enough, it looks fine, and you leave it out long enough, what happens? It putrefies. And if you go look at it microscopically, you see all those, you can see maggots and stuff like that, but you'll see it teeming with microbes. So they theorized, oh, those microbes are arising spontaneously from non-living matter. Okay. So those scientists that were against that tried some experiments. They would take a flask and they would boil the nutrient material, and then they would put a cap on it so that the air you couldn't get air contamination, from the invisible things in the air. And so it would not putrefy. Well, the proponents, those that believed in spontaneous generation, said what you did when you put that cap on that flask is you're preventing some essence in the air that's needed for spontaneous generation. Not something alive, but some essence, whatever, oxygen or some invisible gas or something. So they argued that that did not disprove it. So that's why Pasteur had developed a way to disprove spontaneous generation. And he used heat, okay? And he used something called a gooseneck flask. And put the two together, he used heat for sterilization. He used the gooseneck flask that prevented air contamination back into the nutrient material, and was able to disprove spontaneous generation. I'll show you that in just a minute. But as a result of his work, he developed the laboratory techniques through the use of heat, sterilization, the awareness of aseptic techniques, those techniques we use to protect ourselves, keep our cultures pure, keep us from infecting each other with our cultures, and so on. So in resolving spontaneous generation, he really began the development of some of the techniques we needed. And more importantly, Robert Koch grows the pure culture of the anthrax microorganism. He developed the ability to grow microorganisms in the laboratory in pure culture. Because if he was going to prove that a microorganism was a specific cause of a disease, he would have to have it free of other organisms. If you take a sample from an individual who has a disease, 10 to the 14th bacteria in your body, you know you don't have that disease producer in pure culture. It's going to be mixed in with all those other bacteria, okay? So you've got to find a way that you can separate it and then inoculate that pure culture, that only one type of microbe, into a model animal or into a human and show it causes disease. Because if you just took the sample, you took a sample of blood from a human that had a disease, okay, well, blood might be bad. Let's say some mucous membrane tissue, because blood can be fairly sterile if you're healthy. Okay, and you inoculate it to another human, you say, well, it's a whole multitude of microbes. We don't know which one is actually causing the disease. Okay, so what do we need to develop microbiology as a science? In fact, it developed out of those studies in the late 1800s. The latter part of the 19th century is when microbiology began to be developed. Because at that time, we developed the compound microscope. So we needed microscopy, but not the simple microscope of Hooke and von Leeuwenhoek. Those little single lens microscopes weren't good enough. We needed at least a compound microscope. That happened in the latter part of the 19th century, in the 1850 on. Remember, the Pasteur's work was in 1864, but there, once again, it's after 1850. He developed the sterilization and aseptic techniques. And it came out of his work disproving spontaneous generation. And then the last thing we need was pure culture methods. That could, in other words, the ability to cultivate microorganisms in the laboratory, to separate them from one another, grow them in pure cultures, so he could implicate a microorganism as the causative agent of infectious disease. Now I'm going to just show you some pictures here for a minute. This is Hooke's microscope. It's a single lens system. We've only got one little lens in there. He was able to see cork cells and cork, and he eventually was able to see moles, but no bacteria. They were too small. Oops, I went the wrong direction. Okay. This is von Leeuwenhoek's microscope, and somebody's going to try to give you a bigger picture,